Hello, my name is Zidong Zhang. I'm a recently graduated PhD student from Princeton University. Today, I'll share our work on developing a computational framework for studying gene regulation mechanisms from single-cell multi-omics assays. From our prior knowledge, we already know the existence of multiple mechanisms involved in the regulation of gene expression. As illustrated in this schematic figure, there are cis-regulatory elements on the chromatin such as promoters and enhancers, and there are transcription factors binding to these regulatory elements and regulate gene expression. Our goal is to understand the mechanisms of regulation for each gene in a data-driven manner by analyzing large datasets of different types of genome-wide assays. This data-driven approach has become more and more powerful recently because of the development of single-cell sequencing assays. Specifically, there have been computational methods developed for analyzing gene regulation from different types of single-cell assays. For example, from single-cell RNA-seq, we can link transcription factors to genes based on the co-expression patterns of them across thousands of cells. Similarly, from single-cell ATAC-seq, we can run co-accessibility analysis, and regions that are co-accessible with promoters are probably regulatory regions such as enhancers. However, we think there are still some limitations in these methods. First, each of these methods only uses one type of single-cell assay, and it's not easy to combine them. Second, RNA-seq-based methods usually only look at transcription factors acting on the promoter region and don't look at TFs acting on distal regulatory regions, such as enhancers. Fortunately, on the experimental side, there is a recent development of single-cell multi-omics assay that can run RNA-seq and ATAC-seq from the same single cell, and it provided new opportunities for studying gene regulation more comprehensively. So we developed a new computational framework specifically for studying gene regulation from single-cell multi-omics assays. We call it CREMA, which stands for Control of Regulation Extracted from Multi-omics Assays. The goal of CREMA is to combine the same cell measurements of RNA and ATAC to extract mechanisms of gene regulation with high quality. The input data to the framework is single-cell multi-omics data, where RNA-seq and ATAC-seq are simultaneously measured in the same cell. We first select candidate regulatory regions by looking at the ATAC modality. The candidate regions include both the proximal and the co-accessible distal regions. We also use motif analysis to find potential binding sites of TFs in these regions. Then, in the model building step, we combine the same cell measurements of RNA and ATAC. Specifically, we use a linear model where the regressives are the product of the TF's RNA level and the chromatin accessibility of the TF's binding site. The purpose is to select TFs that not only co-express with the target gene, but also has accessible binding sites in the regulatory regions. In this schematic example, although TF3 co-expresses with the target gene on the RNA level, the binding site of TF3 is not open in cells that highly express the target gene, so we don't include TF3 as a regulator for the target gene. The output from the framework includes regulatory networks connecting TFs to targets, as well as the regulatory domains or regions where these TFs are acting on, including distal regulatory regions. To evaluate the performance of the framework, we first used public databases for evaluation. Here I'm showing one example of the evaluation. In this example, we evaluated CREMA by checking the enrichment of eQTLs in the identified regulatory regions, because eQTLs indicate locations on the chromatin that might affect gene expression. We compared CREMA to a baseline of directly using the co-accessible ATAC peaks using only the ATAC modality of the multi-ohm data. we found that by combining the RNA and ATAC modality in the multi-omic data, CREMA has better performance than method that only uses the ATAC modality. This is using statistically associated eQTLs. We also checked the eQTLs from 3 fine mapping methods reported in the GTEx database to further show that our method successfully identified regulatory regions. Besides using public databases, we also ran the CREMA framework on some in-house generated single-cell multi-omic datasets for the purpose of both evaluation and understanding new biology. In the talk today, I'll share some results from one specific biological tissue that we're interested in. The tissue is pituitary gland. Pituitary is an endocrine gland located under the hypothalamus in the brain. It receives signals from the hypothalamus and releases multiple kinds of hormones into the blood. There are multiple hormone-producing cell types in the pituitary, indicated by the colors here, and they each respond to a certain signal from the hypothalamus and release certain types of hormone into the blood, targeting certain organs and regulate their physiological functions. There are also non-endocrine cells, such as stem cells in the pituitary, that function in reproducing these hormone-producing cells. Zooming out of this detailed anatomy of pituitary, in the big picture of understanding the physiology of whole body and building an atlas of the human cells, it's important to look at the pituitary gland since it regulates the functions of many organs. We first evaluated CREMA's reconstruction of the regulatory network for the pituitary tissue. The way we evaluate is measuring how well the regulatory networks recapitulate gene co-expression patterns in various independent replicates of pituitary samples. The idea is that if the TF-target relation is real, we are likely to observe the co-expression between the TF and the target across many independent datasets under various contexts. As a comparison, we compared CREMA to CINIC, a state-of-the-art method that uses single-cell RNA-seq to reconstruct regulatory networks. We showed that by combining the simultaneous measurements of ATAC-seq and RNA-seq, we have better performance in reconstructing regulatory networks than methods that uses only the RNA-seq. We also want to directly validate some of CREMA's findings, and here is one example. We pulled out CREMA's identification of target genes regulated by the TF GATA2 and the regulatory regions that GATA2 are likely to bind to. We then compared mice with GATA2 knockout to wild-type mice and used the differentially expressed genes and differentially accessible peaks to check CREMA's predictions for GATA2. We found that CREMA successfully identified target genes in regulatory sites of the transcription factor GATA2. We also went on to use CREMA to help us better understand the gene expression patterns in the pituitary tissue. Here is an example about the transcription factor that we're interested in, called FOXP2. Originally, in other fields of research, FOXP2 was found to be involved in speech and language recognition. Later, it was also found to be cancer-related. In the pituitary tissue, we found that FOXP2 is highly expressed in multiple cell types, so we were interested in the regulatory function of FOXP2 in the pituitary tissue. We first used CREMA to find the target genes that are likely regulated by FOXP2. Within these target genes, we found many genes that are previous non-cell type marker genes in the pituitary tissue. Here are some examples of genes that are marker genes for a cell type called gonadotrope. This is also one of the cell types that FOXP2 is highly expressed. When we further examined the expression patterns of all the genes predicted to be regulated by FOXP2, we found something quite interesting. Many of the target genes are differentially expressed between the cell types where FOXP2 is highly expressed. In other words, FOXP2 activated different genes in different cell types. To understand how this is achieved, considering that each gene might be regulated by other TFs besides FOXP2, we constructed the full regulatory network for these genes, and it is shown in this heat map. In the heat map, each column represents a target gene of FOXP2. On the top, the color code shows which cell type this gene is highly expressed in. The rows shows the co-regulatory TFs, besides FOXP2, that regulate these genes. The dark color in the heat map mark the regulatory relation predicted by CREMA. For example, here we highlight a column, which is a gene called FSH-beta. We see that besides being regulated by FOXP2 as shown by the first row, there are some other TFs regulating this gene, such as GATA2 and FLUX5, indicated by the red arrows. Looking at the big picture of this heat map, we notice that although these genes share a common TF FOXP2, genes that highly expressed in gonadotrope cells and stem cells are regulated by different sets of other co-regulatory TFs. We also notice some cluster-like structures in the heat map, indicating a set of genes regulated by a common set of TFs. We did find gold terms and pathways enriched in these clusters that might be interesting for further study. In summary, we developed CREMA, a general computational framework for inferring gene expression regulation from single-cell multi-omics assays. CREMA's identification of regulatory TFs and domains were evaluated by experimental data. We used CREMA to understand the regulatory map of genes with cell type specific expression patterns in the pituitary tissue. For future directions and extensions, we think it will be interesting to apply more complex statistical frameworks that better capture the distribution of single-cell data and the complex quantitative relations between regulators and target genes. It will also be interesting to incorporate new TF binding prediction tools, such as deep learning-based sequence models. It will also be interesting to combine the analysis of gene regulation with the analysis of trajectories and velocities in dynamic single-cell datasets. With that, I'd like to thank my advisor, Olga Transkaya, and every member in the Transkaya lab for discussion. I'd like to thank my collaborators, Stuart Silfon, for organizing these projects, and Frederick for providing high-quality pituitary single-cell datasets. Thanks for attending, and I welcome any questions.